everybody. I welcome you on the second day of this conference. I'm sure we are going to enjoy a lot of interesting lectures today. So I would like to invite the first speaker of the day, Dr. Ila Hawass, please.
Thank you. Good morning. Today I will present the obtained results about the distribution and behavior feeding of Coleophora perplexella. This insect is a pest cereal insect that can affect many species of cereal. This insect belongs to the family of Coleophoridae. Their larvae are protected in cases, and the adult is a Microlepidoptera. We can find in North Africa, Asia, and Europe. Their preferred plants belong to the family of Caryophyllaceae, Chenopodiaceae, Asteraceae, Lamiaceae, and Fabaceae. We can count about 50 species grouped in 5 genus. In Tunisia, and according to the literature, there is about 57 species. The most represented are Coleophora perbera in the northeast of Tunisia, Coleophora guerraciala in southwest, and Coleophora tunisia in northwest. But this species didn't damage any crops. However, during this last year, we remark the very damage on cereal plots. The leaves are dried and died after the attack of larvae of Coleophoridae. This larvae, the identification of this larvae reported that this is a Coleophora perplexella. This insect didn't exist in Tunisia, and according to the literature, it didn't cause any damage. This insect is originated of Spain, Portugal, without causing any damage on crops. It was reported on Hordeum, Murinum, and Bromysterilis in Toulon, France, and in Sardinia, Algeria, and Morocco without identification of the host plant. In Tunisia is a new cereal pest that can threaten national productivity. Some information on the biology of this insect are collected in 2019, but we have started also the distribution of this insect from 2015. So in 2015, between 2015 and 2016, we have reported just one plot attacked with Coleophora perplexella. The next year, there is 4 plots are attacked by this insect. Between 2018 and 2019, there is more other 4 plots are infested by these insects. The next year, we have reported just 1 plot attacked by these insects. Between 2020 and 21, we cannot perform any work about the distribution of this insect because the COVID epidemic. But in 2022-2023, we have reported the presence of 28 plots attacked by these insects. According to this investigation, we reported that this insect infested the barley crops, and with a percentage of 65%, followed by the oat and wheat. So, can Coleophora perplexella infest any other cereal species? In response to this question, we have performed a test on 4 cereal species, such as barley, represented by the variety of Kunuz and Raihan, soft wheat, presented by the variety of Utik and Hydra, durum wheat, variety of Raziq and Karim, and oat is represented by Al-Alia and Fritisa variety. These all varieties are local variety. So, 50 case beers were introduced in not-blowed bots. Each one was surrounded by 8 varieties, the 8 varieties. For each variety, 3 repetitions was conducted. The results obtained about the feeding behavior of this insect reported that this insect reported that the most visited cereal crops are the oat variety Fritisa, followed by barley variety Raihan and oat variety Al-Alia. So, the damage, we have calculated the damage of the diameter of the damage caused by this insect, and we have noted that the case bearers take the largest amount of food from oat variety, Fritisa and Al-Alia. So, according to these results, we noted that an extension of the distribution area of Coleophora perplexella noted from 2015 to 2023, 2 species recognized for their cereal production were attacked by Coleophora perplexella, and the most attacked cereal species were oat and barley. So Coleophora perplexella prefers to feed on oat variety Fritisa and Al-Alia. So to continue this work, we need to perform deep studies on the pest biological and ecological particularities in the order to establish a control strategy. We need to study the effect of different agronomic scenarios, and bioclimatic zone on its kinetic and abundance, you have to investigate the climate change impact on pest insect distribution and ecology, and to explore its natural enemy for effective, sustainable management. This is the first result obtained about these insects, and we will continue working on it because there is no publication or any work performed in this insect. Thank you for attention.
Thank you very much. Any question? It is very scary when a new bug shows up. I was intrigued that the first observation of this infestation was not very close to the seaside. So do you have any scenario how, what's the main route from migration from Portugal or Spain to Tunisia?
Until now, we don't know how can this insect become in Tunisia. Maybe by the commercialization, or maybe with grains. So this is another work to perform, to continue about how this insect is installed in Tunisia.
How far can they travel when they fly?
I don't know.
Interesting work. So we'll switch to the next talk. Thank you very much.
Thank you.
I would like to invite Dr. Mariana Petkova.
Morning. I would like to apologize because I sent my presentation as a poster, but I found out that this morning I have to present, and this is my poster. And the work group from the Agriculture University of Plovdiv has done in determination of the bacteria community in soils associated with the rare wild leguminous species Cicer mombreti and Lupinus albus in Stranja National Park. The territory of National Park Stranja falls into Mediterranean soil area in Europe, and the park is located in Stranja provinces near to the Black Sea coast and very close to the Turkish border. And because this area never fall into ice age in the Tertiary era, there are some species that unique for Bulgaria and all over the world. And this is the location of distribution of yellow podzolic and red soils. And 2 wild leguminous species were found in that area. Was generally considered that Balkans are not the motherland of wild leguminous, but recently we found these 2 species, Cicermum breti and Lupinus albus, which are endemic for Stranja Mountain, and the most important is that the seeds doesn't grow in the other type of soil. We run some physical-chemical characteristic of soil and metagenomic analysis to explain the connection between microbial communities in the plants and in the soil, in a rhizospheric soil, and to find out the connection between local distribution of these wild species and their requirements for soils and special microorganism. And soils were very rich in ammonium, very poor in phosphorus, and rich in kalium, like a kalium oxide. Microbiological analysis done by next generation sequencing, we found out that in red soils there are dominant species. We belong to gamma proteobacteria, which are involved in nitrogen fixation, and in Azotobacter and some Azospirillum, and in the other red soils, predominant species were Actinomycetes, belong to Frankia genus. And the rarefaction curves, this is some ecological parameters of the microbial diversity on the figure 3. When this scale is flat, this is mean that microorganisms are in biological equilibrium. And when it's sharp, this is mean that they are not in this equilibrium. And there is almost no very big difference between the 2 types of soils. The species community are in equilibrium according to our results. Indices of diversity and beta diversity. Alpha diversity, it's an ecological index, and it's calculated by 5 indices: Shannon diversity, Simpson diversity, Chow's diversity, and it gives the picture how a community is the situation, the relationship between different members of the microbial community. And indices, Shannon, more than 8, it means the very good index for ecological equilibrium in this system. This is mean that these soils are very good ratio between and relationship between the microorganism. And the next picture is the vein diagram. And this vein diagram, this is picture number 4. The result shows how that almost 124 species are common for all the this type of soils, but we have a unique microbes which give the differences between these soils. And beta diversity on the heat map. It gives the relationship between two. We can compare two different type of microbial community relationship in two different soils. And by ratio, we can check the diversity, how the diversity of microorganism is connected with the soil fertility. And with you can see that the cinnamon soils that are found in old oak forest are with a better beta diversity, and this is comparing with the red sandy soils. And finally, we've done the plot. This is the principal component analysis. On this principal component analysis, the closest results, this means that soils has common conditions on and microbial diversity, and when they're distant, more than 60% distance between the red soils and cinnamon soils. This is mean that cinnamon soils that are home for the Cicel mombreti wild species are with a unique microbial diversity, but red soils are distant 60% from them, and their soils that we found, Lupinus album, again, I said this is the species that are located only in Strandzha Mountain, endemic to Strandzha Mountain. And that this is we have to done a lot of things further to examine the soils, like physical-chemical characteristics, and to explain this local distribution, because we tried to grow Cicel mombreti in Chernozem's soils, but we didn't succeed, or in vitro techniques, we also didn't succeed. It grows only in that soils, and we will try to microbiological, physical, chemical properties of soils to explain this local distribution. Thank you very much for your attention.
Thank you very much. Again, very nice talk, despite the technical problems.
Thank you.
Is there any question? Yes, please.
Thank you very much for your presentation. It's really interesting. And my question is connected with the physical-chemical characteristic about the soils. Is there some influence on the pH level of these soils on the microorganism associations?
Yes, normally Lupinus species, by literature review, they grow in acid, less than 5 pH. And really, distribution in red soils, the pH of soil was 5.8. Because they grow in this old oak forest, and especially in fall when the leaves are decomposing, pH even drop. But yes, we try to make a similar condition by adjusting pH of media, culture media, and even with the soil, experimental soil in the lab, but we couldn't succeed to grow the species in the lab or in the laboratory fields. Only in Strandzha Mountain they grow.
I have any other question? I would like a clarification, a technical detail. When you do the sampling, do you take soil sample anywhere on the field or close to the root of a certain plant?
Exactly, because for DNA analysis, NGS analysis, next generation sequencing analysis, we extract DNA. And we subject this DNA for 16S identification of bacterial and fungal endophytes and rhizospheric. Exactly, we are washing the soil that has it to this root, rhizospheric soil. Not, of course, we've done in 10 and 20 centimeters far from the plants. We also analyze the soil. We get many soil samples but it's local distribution and plants are not so widespread. Only especially Cicermum breti.
Thank you very much. If there is no other question, we thank you very much.
Thank you.
We move to the next talk. This is from Dr. Anbar Ahmed from Pakistan. So the next speaker did not show up, so we'll have an early coffee break now. Thank you very much.
University of Thrace in Greece, and honored to conduct this session. I will welcome all the participants. Thank you for your presence. I'm very glad to introduce the invited speaker of this session, my colleague, Professor Rafael Tsanaltsopoulos. He's coming from the same department, the Department of Molecular Biology and Genetics of our university. He specializes in molecular biology and particularly gene expression and signaling mechanisms, and then with emphasis on the tumor microenvironment of cancer. His speech entitled "The Road Towards the Adoption of Biosimilar Recombinant Polypeptide Drugs, Socioeconomic Factors and Genetic Engineering."
Thank you very much, Dr. Tokatlidis. Değerli konuklar, merhabalar. It is a pleasure to be here in Edirne because my grandfather was born here, and it's very nice to be with you here. I should thank the organizing committee for giving me the pleasure for the invitation. And today I'm going to discuss with you a paradox. The paradox is that we have very expensive prototype drugs available, and we also have quite inexpensive biosimilar drugs that are equally effective and equally safe, but consumers still prefer to spend a lot of money and buy the prototypes. And I'll try to understand that and discuss with you the situation in the Greek market, and I'm sure the situation is similar in many other countries, and maybe exchange ideas. So, what are biosimilar drugs? Biosimilar are drugs that are very similar to the original drug. They have exactly the same active compound, and they have been tested, and it has been shown that they have exactly the same therapeutic outcome when they are administered at the same dose to the patient. But biosimilars are allowed to enter the market only after the expiration of the patent that covers and protects the original prototype drug. When they enter the market, they either have an international non-proprietary name, for example, paracetamol, or they have a branded name, they are called branded generic when they are given a brand name. Biosimilars are used in healthcare systems worldwide as an effective alternative to much more expensive original drugs. An original drug may be produced only by the company that has invented it and has developed this drug. So original drugs are produced by only a single pharmaceutical company, while many different biosimilars may be produced by different companies. The procedure to get permission to enter the market for a biosimilar is much, much easier and much, much faster and cheaper than the prototype. Therefore, the cost at the end is much lower. At the end, biosimilars may be sold at a fraction of the price of an original. Still, biosimilar production may offer a great profit to the industries that produce them because the cost of development is very low, it's minimal, and they can save the budget of the healthcare system. However, the 1 million question is, given that the biosimilar drugs are equally effective for patient treatment and far more inexpensive, why aren't they massively adopted by the market? Why consumers still prefer the original, more expensive drugs? And there are a lot of socio-economic factors that lead to this decision, this behavior. I'm trying to analyze that using Greece as a case study. So let's see the Greek market for drugs. There are 10,427 retail shops, pharmacies, point of retail sales. There are 106 drug manufacturers or drug importers in Greece and 136 drug wholesalers. So this number, we have to evaluate it if it's high or low. Let's compare it with other European countries. For example, if you compare it to France, France has 20,000 retail points. It seems maybe okay. But if we take into account that France is about 6 to 7 times greater than Greece in population, then we can scale it, we can calibrate for per 100,000 people, and we can see that although the EU average is 32 points of sale, Greece has 96, 3 times more. France is very close to the average. So the first important observation is that Greece has way too many retail points. And this number is stable across many years, traditionally, in Greece. The number of wholesale distributors is also pretty stable along the years. So why do you think a great number of retail points is important for, is an important obstacle for the entry of biosimilars? One could think that, okay, if you have many retail points, you don't have collusion, you don't have price fixing. But in the drug market, the prices are fixed by the government anyway. The pharmacies, when they have so great number, they try to sell the most expensive drugs, so they have a greater profit. So that's one thing we have to take into account. Greece has a very strong drug production industry. 34% of drugs that are sold in Greece are being produced in Greece, they are locally produced, and the other 14% are locally produced with a license from the proprietary. So there is strong drug production capabilities in Greece. In certified facilities, there are 28 big factories, drug-producing factories, with very high quality standards. So the high price is not dependent on the availability of production capacity. Indeed, the number of people who work in drug industry in Greece is ever increasing. There is a 55% increase since 2015. And this number, compared to other European countries, is very high. 7% of the total industrial employees are working on the drug-producing sector in Greece. So it's much higher. It's actually double than the average of the EU 27. The annual drug sales in Greece in value is 7 billion euros. 4.7 out of that are sold by retail, by pharmacies. The important thing is that out of this 7 billion, only 36% are biosimilars. And this is extremely low. It's the lowest among European countries. The average in Europe is 62%. We have 32% off-patent and 8.5 on-patent prototypes. So in total, about 41% prototypes. So why Greeks are spending so much money to buy ridiculously expensive drugs when they have the option to buy an extremely cheap biosimilar? That's the big question. And please note that there is a striking negative correlation between the price of original drugs and the volume of sales of biosimilar. This means that when you have when you have a small percentage of biosimilars in the market, then the price of the prototypes goes up. So we buy more and ever more expensive drugs in Greece. There is a good trend, though. Over the years, we increased to buy more and more packages of biosimilars compared to original. The problem is that the price of biosimilars is so low compared to the original that the percentage of the market share of biosimilar is still going down compared to 2014 as a base year. So we're not in a very good position. Okay, we are talking about billions to the total economy of Greece. What happens per capita? Every Greek, on average, spends about 500 euros per year for pharmaceuticals. This is a ridiculously high number of money, high amount of money, especially if we compare Greeks with Cypriots, for example, because they have similar genetic background, so one would expect to have similar diseases, etc. So in Cyprus, per capita, they spend about half the money that one spends in Greece. Okay? And so this means that we have an overconsumption of drugs and actually an overconsumption of very expensive drugs. So it's a kind of bad habit. Especially interesting is that in Cyprus, 85% of this expenditure is spent by the government, and only 14%, actually 10%, is out of the pocket payments. While in Greece, 45% of this expenditure is paid by out of pocket. It's paid by the patients themselves. Okay, so this is a paradox. The proportion of private burden is very high in Greece relatively to other countries. So the conclusion is that there is a need for implementation of new national policies and strategies that will alter consumers' behavior and attitude. In order to understand how much this cost is, I have put here a number. Almost 10% of the gross domestic product is an expense that goes for drugs, for healthcare, and 9.7 of that is government spending, and the rest, about 6 billion per year, is spent by consumers, out-of-pocket money. This is a lot for the standards of Greece. So the conclusions and proposals is that there is significant overconsumption of drugs in Greece. So the proposal is that we have to implement immediately drastic strategies to modify consumer behavior. The consumption of biosimilars is relatively restricted compared to other countries, so we have to adopt policies to boost the market share of biosimilars. And the Greek pharma industry is robust and has the potential to produce biosimilar drugs, but we have to create suitable conditions and maybe give incentives to strengthen biosimilar drugs production in Greece. Now, I'll give you an example. India was about to reach the point of complete crash of the health system because of the cost of polypeptide drugs, mainly insulin analogs. The cost per patient was over the roof, and the government made a very bold decision. They said, okay, we will not allow any prototypes, and they offered incentives to local industries to develop biosimilars, and now the cost of the treatment for insulin requiring patients dropped by 100-fold. That actually almost saved the healthcare system in India. Things like that should be, strategies like that should be taken also in European countries, but Europe is much more open market, so you have to create other kind of strategies. What strategies you can take? You have to find out all the little triggers that you have to give to doctors, the pharmacists, and the patients in order to work better together. And you have to change the regulatory framework. You have to educate consumer, and you have to implement marketing campaigns that lead consumers to prefer biosimilars. So this concludes the first part of my talk. The second part, I'm going to focus on some efforts in our lab to develop biosimilar drugs in collaboration with other laboratories in Greece and also a pharmaceutical company in Greece. And the case that I will present today is the production of interferon alpha-2A. It's a cytokine, a signaling protein, and I'll talk about the expression, purification, preclinical analysis of biosimilar polypeptide drugs with improved characteristics. So our aim is not only to produce a biosimilar, an equivalent to the prototype, but a protein that is even better, that has better characteristics than the prototype. So interferon alpha-2A is a secreted glycoprotein. It's a small protein, only 165 amino acids, that is organized in a very nice spherical structure comprising 5 alpha helices. It's actually quite robust, very stable, and very soluble, so it's pretty easy to express and work with it. It has anti-oncogenic and immunosuppressive activity, and it is administered in many situations, mainly in patients with viral infections, persistent viral infections, such as hepatitis and HIV, etc. The most common drug form in the market is lyophilized powder that you reconstitute and then you inject to the patient. Interferon is not really a global blockbuster drug, but it's pretty essential. The market is between $500 and $1 billion US dollars global. So it is worth to work with this. Interestingly, the patent expired 8 years ago, and the patent cover Roferon A, it's the human sequence for interferon alpha-2A from a company called Roche. And today, the most important player in the market is Pegasus. The aim of our project was to express this protein in a secreted form. Why we wanted to express it in a secreted form? Because it's much easier to purify it, because you don't mess with an extract from homogenized cells. You just take the supernatant of the culture. You have better chances for native folding of the polypeptide, and this, in turn, guarantees improved biological activity, and I'll show you some comparisons later. And also, it may furnish the protein with post-translational modifications, because the protein, through its way for secretion, it goes through the Golgi system. Therefore, it can be glycosylated, and this protein also has some O sugars. And once we purify this protein. We want to develop a superior drug form. And I'll talk about the Hitosain-based gels later. I will not cover microsphere encapsulation today. The idea is that by putting the protein in a capsule, you can achieve a delayed release in the bloodstream. And that's very important because you don't have a sudden surge, a very high peak in the circulation, but you have a constant and delayed release that lasts much longer. The system we used, Pichia pastoris, is a methylotrophic yeast. It uses alcohol oxidase. We use the alcohol oxidase regulator system. It's a promoter that is very strong and is induced when you add methanol in the culture. And it allows, it has very low secretion of endogenous gene products. So the majority of the proteins in the supernatant of the culture is the protein that we cloned. We use this vector, pink alpha, high copy. This is important because it boosts the production, higher levels. We cloned this position under the control of the AOX1 promoter, and we cloned the sequence encoding the human polypeptide using the codon bias from Pichia pastoris, and on the 5' end, the N terminus of the polypeptide, we encoded 2 affinity tags and the sequence that is recognized by a peptidase, specific peptidase that we can use to release this and then free the native full intact protein. So the methodology is, once we clone that and we sequence and do all the controls, we transformed and selected these big white colonies here in the selection medium. We grow in glycerol medium. In this medium, this gene is not expressed because we don't have any methanol. It's very tight negative regulation. And once the biomass is increased, then we add methanol and we start in large-scale defermentation, and therefore the secretion of interferon in the supernatant. Then we affinity purify the recombinant secreted polypeptide. We cleave the affinity tag and we release the purified folded recombinant human interferon alpha 2A. This is the purity we get. This is a Western, the full length, and this is after the release of the affinity tag. And then we continued first with the investigation of antigenicity and immunogenicity. You don't want a polypeptide that you intend to inject in humans. You don't want to trigger antigenic immune response. So we tested it. We did a very thorough in silico analysis, but I won't talk about that. We detected antigenicity with ELISA assays, and we checked whether, with FACS analysis, whether the addition of our recombinant protein triggers release of cytokines from T cells from the blood samples of healthy donors. So the antigenicity of our polypeptide is very similar, actually it's identical to the FDA-approved available protein in the market. And importantly, here you can see in a FACS analysis, these plots. Here we counted in single cells the expression of 4 different cytokines as a result of the addition of our protein. So for those of you who are not very familiar with this kind of plots, if you see here, this is the positive example, the positive sample, where we stimulated we stimulated these T cells. You see this shift to the right, and then in the other samples, importantly, the gray ones and the dark green ones here and here, you don't see any shift to the right. Even if you add twice the amount of the normal dose of interferon, you don't have any extra immunogenicity, and that's very good. Then we wanted to evaluate the structural stability of our protein. The first thing we did is a CD spectrum. CD spectrum, from this spectrum one can deduce the amount of secondary structure, how many alpha helices you have, and we compared our spectrum with the spectrum previously published of the native protein. They are identical, and that was a very good result. And we then tested its stability as a function of increasing temperature. We used differential scanning fluorometry for this. This technique, we use a fluorescent dye that fluoresces only when it binds to a hydrophobic surface of a protein. So when our protein is soluble, there are no hydrophobic surfaces, so the fluorescence is very low. If we increase the temperature a lot, then the protein denatures, it unfolds, and it exposes hydrophobic surfaces, and therefore the label, the fluorescent dye, contacts these lipophilic surfaces and becomes fluorescent, and therefore the signal increases. So when we studied our protein with the red line here, we see that it is stable pretty much above 60 degrees, and the graph is exactly identical to FDA-approved interferon that we bought from the market. Okay, so our protein is very stable. And then we wanted to check the biological activity. So what we did is we used cell culture, actually several cell cultures. I'll show you two today. This is from lung cancer cells, where we added either 1 nanogram or 10 nanograms of our Pichia pastoris expressed protein. The cells look identical to untreated, and the same is right for FDA-approved interferon. But when we measure the levels of interferon alpha-2A target genes, we measure the mRNA levels, we see that when we add our protein, the signal increases, and actually it always increases higher than when we add the same amount of the FDA-approved over-the-counter drug. So the biological activity of our protein is superior to the one of the prototype. Okay, and please note that this is not a linear, it's exponential scale, so these differences are pretty robust. Okay, and this happens to all of these target genes. We tested several target genes, and it's also dose dependent. And here is another cell line. This is very, very repetitive. So the last thing I'll discuss is the superior drug forms. We created a hydrogel based formulation that allows salt gel transition at body temperature. The idea is that you have your drug in solution, you inject it, and when it comes in the body at 37 degrees in body temperature, it solidifies and it's a gel, and then it is gradually released in the circulation. We achieve that with a polymer called Hitozane. It's derived upon deacetylation of heating, and we have an equilibrium with beta-glycerol phosphate, and upon transition from solution to gel, when you shift temperature, you create a meshwork. In scanning electron microscopy, it looks like that, and the drug polypeptide is trapped in these cavities, and then it's released slowly, slowly. And this is the kinetics of the release. So instead of giving all the drug immediately when you inject and then you have a localized depot of the drug that is released in circulation slowly. So the conclusions, to summarize the conclusions, interferon alpha-2A was successfully expressed and purified from Pichia pastoris. The recombinant protein maintained structural stability. It retained its antigenic epitopes, and significantly, it did not induce T-cell activation in pilot in vitro experiments. It showed a considerably higher biological activity compared to marketed bacterially FDA-approved counterparts, and Hitosan-based hydrogels may be used to delay drug release in circulation. Actually, at the moment, we have a series of experiments with mice, and the slower release has been confirmed also in vivo. And I would like to thank my colleagues at the Department of Molecular Biology, faculty members, Katerina Chlichiagi, Papa Alexis Galanis, Adonis Giannakakis, Vicky Sfadoulou, and Nikos Glikos, all the students in the lab, mainly Sophia, she's a PhD candidate that did most of the work, the expression purification, Maria Tokamani, Katerina Spiridopoulou did the fax analysis, and Elias Chokadaris here did all the work with the cells, etc. And George Triadafilidis is special counseling, who helped me a lot with the financial analysis. And this is a work co-funded by Greek and European funds. It's a collaboration with the University of Patras, Department of Medicine, Department of Pharmacy, and the company called Verisfield. It's a pharmaceutical company in Greece. Thank you very much.
Thank you, Rafael, for this excellent talk. Thank you also for being within the time. We have a little time for questions. Please.
So thank you very much for the nice talk. I have one question. Have you also did some side effects experiments with these purified IFNs?
You mean in clinical trials,
yes.
in patients?
Yes. In patients or in mice? Sometimes they do it also in mice?
The experiments in mice last only a few weeks. So we wouldn't see any side effects there. We are lucky that this protein is exactly the same in human and mice, so we don't have any immunological response in mice. They tolerate the injections very well. They look healthy, but we keep them only for a few months. These drugs are used for years and even decades normally in humans. If it's exactly the same protein, we don't expect any.
No, but I mean, if you want to use those drugs, and you have to somehow to show that it has no side effects and it is the same. So the people, they want to see it.
So the procedure in order to get a license for a biosimilar is after you complete all the lab experiments, then you have to find volunteers. You give them some incentives, and they try this new drug, and you scrutinize, you monitor them to see if there are any side effects, any abnormalities. And after, then you go from a limited number to a greater number, etc.
So the clinical trial.
We haven't reached that point yet.
right.
We haven't
But
reached that point.
very
but
nice project.
Yeah.
Really, I
Thank
like it. Yes. And can you also do or produce other drugs with
actually, in this,
more, which are larger, for example, larger amino acids?
larger proteins,
Yes,
you mean
large-
longer
yes.
amino
Yes.
acid chains.
In these yeasts, is it
We
possible?
haven't tried much longer. We have tried two other proteins, erythropoietin alpha.
Yeah.
That was not very successful because it had
Ah.
too much glycosylation,
Ah, okay. Modification.
abnormally much, so
Yes.
we abandoned that.
Yeah.
And we had another protein, IL-2,
Yeah.
a form of IL-2,
Mm-hmm.
that worked pretty well. We produced it, but there was a problem in the structure, and
Yeah, that is the problem with the
the
large
problem was
proteins.
there
Yeah.
were that protein has a lot of cysteine, so you can have alternative bisulfide bridges, and you can some molecules can be trapped permanently in a wrong structure.
Yeah.
And when we noticed that, we went and we found out that even the
Small.
prototype, they have mutated several cysteines. So what is in the market is not the human sequence of the protein.
Oh, really?
Probably they had the same problem and
They
they solved
changed
it
it.
by mutating
Yeah.
cysteines.
Right. Thank you very much.
Okay.
Okay, thank you very much, Professor Sangarazopoulos. We now proceed with the second speech by Xuri Iad. Is here speaker. The title: Assisted Extraction Optimization of Bioactive Compounds from Cucurbita maxima by Products using RSM design. No. Proceed in effect. The title: Prospects and Potential of Monica oleifera spp of Pakistan as a natural perspective for granted. By Yamin Pipi. No. So I call the there are two additional speeches added in the schedule. Biology of the blue crab Calip. Biology of the blue crab Calinectes sapidus, invasive species newly introduced in Algeria. Malta Tari.
Ladies and gentlemen, thank you for joining me today for this communication on blue crab, on American blue crab. This topic is of great concern and requires our attention as it has significant ecological and economic implications. The American blue crab, scientifically known as Calinect sapidus, is native to the Atlantic coast of America. However, due to various factors, such as ballast water discharge from ships and the rising of temperature, it has been introduced to European and African countries. This species is known for its aggressive behavior and ability to outcompete the native species for resources, what is happening with the European eel in our region. It can also have negative impacts on local ecosystems by altering food webs and damaging habitat. In the last decennies, different papers reported its arrival in Europe, such as Spain, France, Italy, and even here in Turkey, and also in North Africa, as Morocco, Tunisia, Libya, Egypt, and in Algeria. In Algeria, its first record was in 2019 in the brackish water, and last year's even in fresh water. So, as a scientist, it's important to understand the ecological impacts and finding ways for the maintaining of biodiversity and ecosystem health. To achieve this objective, we sample 100 individuals from November 2021 to June 2022. And we take the different body measurement and we obtain the following results. The sex ratio was in favor of females, and the weight of female was huge, between 100 and 200 gram. The length between 5 and 8 centimeter. The width between 30 and 70. The thickness was between 15 and 21. And the forehead was between 3 centimeters and 6. Contrarily, males were more small. The weight was varied between 58 and 30, more than 300 gram. The length varied between 3 and 8 centimeters. The width varied between 9 and 15 centimeters. The thickness varied between 12 and 20 centimeters, and the forehead between 4 and 6 centimeters. The allometry of the total population, here we find that blue crab in North Africa, in Algeria, have a fast growth. It grows fastly and very well. The female has K equal to 2, and the male have K equal to 3. Finally, our work, the work was carried out between our results, findings, as I say, we find that the blue crab newly introduced in Algeria grows very fastly because they find a good climate, and for more research we should continue the same study in different ecosystem, brackish and freshwater, and we should increase the number of crab fished in order to reach the totally of population, and to be able to establish a comparison between different sizes class. And I thank you for your attention, and feel free to ask me any questions. Thank you.
Okay, thank you very much for this concise speech.
Thank you.
Any questions, please? Okay, thank you.
Thanks.
The next speech is the title, Biodiversity, Bioeducation and Ecology of Epiphytic Lichens on cork oak in the Filfina Forest Massif, northeastern Algeria, by Utabia Lamia.
Mesdames et messieurs, bonjour. Donc, très honoré de vous présenter notre communication qui s'intitule donc Biodiversité et bioindication, ainsi que l'écologie des lichens épiphytes. Alors, c'est une étude sur les lichens, donc les lichens épiphytes, donc concernant leur biodiversité et leur bioindication, ainsi que leur écologie, et ce au niveau d'une forêt ou des forêts de chêne-liège dans la région de Filfila, la forêt donc de Filfila au nord-est algérien de la wilaya de Skikda. Alors, il faut savoir que les lichens sont des bioindicateurs du milieu. Donc, ce sont des végétaux qui sont un petit peu sous-étudiés, donc, notamment en Afrique du Nord. Donc, en Algérie, les études ont été faites depuis, c'est-à-dire le XXe siècle. Ils se sont succédés par d'autres études, donc par des chercheurs nationaux et internationaux. Donc, ces derniers temps, plusieurs régions sont concernées par l'étude des lichens, notamment donc, à l'extrême est donc, algérien, donc, par des travaux de thèse et par des articles donc, de recherche, notamment dans la région d'Al-Qala, donc le parc national d'Al-Qala. Cette étude vient compléter un petit peu ce qui a été fait à Al-Qala et à Annaba. Donc à Annaba par rapport à la péninsule de l'Idour. Nous sommes orientés vers une étude des lichens de Filfila. Donc Filfila qui est un petit peu une copie, c'est-à-dire de la péninsule de l'Idour, parce que c'est une région qui englobe une richesse très importante en plantes, donc surtout les plantes endémiques et aussi les ptéridophytes. Nous avons en 2020 retrouvé une nouvelle espèce de ptéridophyte dans la Numidie, donc à l'est algérien, Pteris vittata. Et là, l'idée est venue pour voir ce qui concerne donc les lichens. Donc il faut savoir que cette étude est effectuée dans le cadre d'une thèse de doctorat, présentée donc, c'est-à-dire faite par notre doctorante et supervisée par moi-même. Donc, le but, en fait, de cette étude, c'est de voir d'abord qu'est-ce qu'il y a comme richesse au niveau de la subeurée de Filfila, et donc un listing pour enrichir le checklist ou la checklist donc, des lichens algériens, mais aussi d'étudier un petit peu le fonctionnement de cet écosystème dans un milieu qui est pas loin d'une source de pollution. Et à travers les lichens, on pourra savoir s'il y a donc, s'il y a des lichens donc indicateurs dans les deux sens, c'est-à-dire indicateurs de pureté d'air ou indicateurs de pollution. Sans oublier aussi, il y a un aspect de changement donc, climatique par rapport à des espèces qui sont très connues donc, au niveau de notre pays et aussi au niveau de la Méditerranée. Donc, Filfila est une région qui se retrouve donc, à l'extrême nord-est algérien, dans la wilaya c'est-à-dire de Skikda. Donc, c'est un massif forestier recouvert par des forêts de chêne-liège ou de subeurée. Alors, le choix des stations, il est, il est en fonction de l'homogénéité de l'habitat, mais surtout de l'importance des phorophytes. Il faut savoir que les phorophytes, ce sont les supports des lichens. Donc, nous avons travaillé sur 20 phorophytes, donc c'est-à-dire la richesse lichenique de 20 phorophytes, donc à travers ce qu'on appelle des relevés systématiques qui sont complétés par la suite par des relevés phytosociologiques. Parce que ce qui nous intéresse aussi, c'est d'établir une liste, mais aussi de faire ressortir les groupements phytosociologiques. Donc des indices écologiques donc, qui sont récemment proposés par professeur Nimis, un éminent lichenologue italien. Donc ces indices écologiques concernent d'abord la catégorie, c'est-à-dire hormis la catégorie physionomique, ça concerne le photobionte, ça concerne la reproduction ou la stratégie de reproduction et aussi les chorotypes. Donc comme résultat, les résultats préliminaires de 2 ans, c'est-à-dire de 2021 et de 2022, s'avèrent très intéressants déjà, parce que nous avons retrouvé déjà 97 espèces réparties en 18 familles. Les espèces ou les familles les plus importantes en nombre d'espèces, ce sont les Parméliacées et les Phyciacées. Et donc, les genres les plus importants par rapport à ces deux familles, ce sont le genre Parmelia et le genre Phycia. Alors, pour la catégorie lichenique, il faut savoir que chez les lichens, il y a 6 types morphologiques, et là, nous avons retrouvé les 6 types. Mais seulement, la catégorie la plus importante en nombre d'espèces, c'est la catégorie des lichens qu'on appelle crustacés ou incrustés, suivie des espèces foliacées. Les squamuleux sont généralement en nombre moins important, mais là, nous avons retrouvé une espèce de lichen squamuleux. Alors, par rapport à la catégorie, ou la stratégie de reproduction, ce sont les espèces sexuelles qui dominent, avec 57 espèces, 57%, suivi des espèces licheniques qui se reproduisent par voie asexuée à travers les sauridies. Vient par la suite la voie asexuée à travers les isidies et en dernier les lichens qui se reproduisent par voie asexuée mais à travers des fragments. Pour ce qui est du photobionte, parce qu'il faut savoir que les lichens c'est une association entre un lichen et un champignon, donc le champignon va donner les éléments minéraux, l'eau et le support, l'algue va donner donc c'est-à-dire la nourriture carbonée. Là, on retrouve que les algues vertes dominent avec force 76%, donc par rapport aux autres espèces donc, telles que les cyanobactéries, donc qu'on retrouve notamment chez les espèces qu'on appelle les gélatineux. Alors, pour ce qui est de la strate de la végétation, donc c'est la strate donc méditerranéenne ou l'étage méditerranéen qui domine, donc suivi du supra-méditerranéen, plutôt suivi du méso-méditerranéen, en troisième position le supra-méditerranéen et en dernier le montagnard donc méditerranéen. Alors, et on termine par le type de chorotype, et comme vous le voyez donc à travers l'histogramme, ce sont les espèces tempérées et les espèces subocéaniques qui dominent par excellence, contrairement donc aux espèces méditerranéennes et les espèces méditerrano-atlantiques. Alors, pour le groupe phytosociologique, donc le deux groupes s'avèrent donc par excellence dominants. C'est la population nitrophile, donc puisqu'on parlait de bioindication, ça veut dire qu'il y a une quantité de nitrate qui s'évapore dans l'air importante, d'où la présence d'espèces donc nitrophiles, et cela revient au fait que nos forêts algériennes, notamment à l'est algérien, subissent ce qu'on appelle le surpâturage, c'est-à-dire que les éleveurs ne font pas leur élevage donc, en intensif, ils le font plutôt en extensif. Et donc là, à travers les bousses de vaches et tout ce qui s'ensuit par rapport aux poussières, viennent s'installer les lichens nitrophiles. Donc, entre autres, l'association lichenique qu'on appelle Physitum ascendentis. Alors, le deuxième groupe, ce sont les populations sciaphiles. Et donc là, ce sont les espèces qui se cachent derrière un petit peu l'ombre et surtout qui se cachent bien à travers, par exemple, la partie du tronc qui se trouve dans une ambiance de végétation, parce qu'il faut savoir que certaines stations, ce ne sont pas des stations où on retrouve uniquement du chien-liège, donc c'est-à-dire à part, mais on retrouve un cortège floristique qui arrive jusqu'à une hauteur de 1 mètre jusqu'à 2 mètres. Et là, dans cette ambiance ou dans ce type de microclimat, va s'installer une certaine humidité qui va favoriser l'installation de ces espèces qu'on appelle donc les lichens sciaphiles, notamment le fameux lichen Perinula chlorospila. Alors, voici quelques images pour les lichens donc, qui sont un petit peu emblématiques de la région. Donc, on retrouve le premier genre, c'est le genre Usnea. C'est un lichen fruticuleux. Donc, et leur présence, par contre, cela indique un petit peu la stabilité de la station ainsi que la pureté de l'air. Vient ensuite les Ramalina, donc, qui sont très importants et qui sont étudiés, donc, même dans le domaine de l'ethnobotanique. Là, vous retrouvez donc, les Ramalina farinacea et les Ramalina fastigiata. Pour ce qui est des lichens crustacés, et la difficulté par rapport aux recherches justement licheniques en Algérie, c'est par rapport à ce groupe-là, parce que c'est un groupe qui, dans l'identification, ne s'arrête pas uniquement à une identification morphologique ou même à une identification à travers, c'est-à-dire un traitement chimique. Là, on est obligé de passer au laboratoire, parce que des fois, l'image vous donne, pour deux espèces, vous donne une idée qu'il s'agit d'une, alors qu'il s'agit d'eux, parce qu'on est obligé de faire des coupes histologiques et de déterminer le nombre de spores. Et c'est ce qui complique un petit peu la chose par rapport aux jeunes chercheurs algériens. Donc là, vous avez le Perinula chlorospila avec cette couleur, donc un vert assez luisant, parce que là, il a plu, et donc c'est un petit peu l'image d'un lichen donc, humidifié, et à côté, un lichen qui est très très connu par rapport aux crustacés, c'est l'Icidella et l'Aochroma. Donc en conclusion, ce que je pourrais avancer dans cette étude, c'est que les résultats préliminaires s'avèrent très intéressants et nous encouragent à aller au-delà de ces données. Et c'est-à-dire, je voulais, à travers donc, cette communication, passer un petit peu le message à mes collègues donc, algériens et même à nos collègues donc, ici en Turquie pour un petit peu échanger, s'il y a des lichinologues ici, échanger donc, nos relations à travers c'est-à-dire ce type de plantes. Donc, je vous remercie pour votre attention et je rends la parole à Monsieur le Président. À vous la parole, Monsieur le Président. Merci pour la parole.
Okay, thank you very much. Any questions? This is the end of this session. I thank again the three speakers. I apologize if I'm sure I mispronounced your names. So now it's time for lunch. Please, we'll be back in about an hour for the next session. Thank you.